Hello everyone. My name is Jan Magielski. I'm the Neuroscience Society Vice President for External Events, and it's my pleasure to welcome Dr. Anderson with us today. Dr. Anderson is a Director of Research for Child and Adolescent Psychiatry at CHOP. He's also Professor of Psychiatry at Perelman Med School, and he also is a Principal Investigator of Anderson at CHOP. So welcome, Dr. Anderson. We are very happy to have you here, and yeah, the floor is yours.
Great, yeah, thanks so much for the invite. I'm happy to be here. My first disclosure is that, as I was saying to Jan, I was in Florida canoeing in the Everglades this past week with my family, and we got stuck behind some bad weather and weren't able to fly out Sunday, and so we flew out this morning, and I had to get up at 3:30 in order to, we had to make the plane. So if I seem a little bleary, that would be like the day after getting a grant done or something. But in this case, is the day after, working our way back. Flew into Trenton Airport for the first time, which was exciting. Yeah. So what I'm hoping we can do is be very interactive, and it would be obviously so much more fun, at least from my perspective, to be in a room, where we can actually see each other and stuff. But I'm hoping that we can be interactive. I'm hoping that people are comfortable just stopping me wherever, and so that we can kind of chat about stuff. I would think you guys would get more out of it, and I think I'd probably be better that way too, rather than me just kind of blah 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 blah, going on about stuff. What I thought I would do first is I'll share my screen and just show a slide from a talk I gave actually at my school, my college, when I went back for the only reunion I've been to was a few years ago, and they asked me to give a talk about research, and I thought I could use this to kind of go a little bit through the development of my own particular way of getting to where I am as a physician scientist. And so kind of the first thing, at least in my development, was that I was always a pretty dorky, science-oriented kid, and I went through phases of wanting to be different types of scientists, like geology and astronomy. And then I read a book about the brain, and I thought that was really interesting compared to anything else in terms of complexity. Maybe not more complex than the astronomy, I suppose, but to me, it just really captured me, and it was so relevant. Something else that just happened to happen to me was one of my friends when I was 17, and this is the late 70s, and in a suburb of Hartford, Connecticut, pretty druggy public school, and one of my friends was a pretty normal kid, and he just suddenly, we didn't really even notice him acting weird. He kind of got quiet for a little while, like a month or something, and then he just kind of disappeared. We didn't really see anything strange happen. And I only found out a couple of years later, by the time I was a freshman in college, that he developed schizophrenia, went through a bunch of hard times, and suicided. And his family completely hid it from everybody. He just disappeared, and nobody really knew where he went or what happened. The family didn't talk about it, and it was only a couple years later I kind of found out what happened. But that also, I was already interested in brain development, but that helped me to understand, like, how could that happen? How could somebody be like pretty normal, and then be like really not normal, and in a way where it's not just, okay, some people suicide, especially adolescents, can do that sometimes when they're they have a breakup or something happens that really upsets them, and people can be kind of spontaneous and do something that they can't take back, like kill themselves. But in the case of schizophrenia, it's usually not so much like that. It's not impulsive. It's usually people are sick, and their biggest time when they tend to hurt themselves isn't when they're the sickest. It's actually when they're better. The biggest risk for suicide is when they leave the hospital and they're less psychotic, hopefully, and hopefully have some kind of social situation arranged for them, maybe going back home, maybe going to other relatives, maybe going to a halfway house or something. But they realize how messed up their lives have become, and it's sort of an existential thing. That's when they're sort of at highest risk for killing themselves. And that's probably what happened to Ricky. So anyway, that caught my attention in a way. I was already interested in brain. I went to Amherst College because back in 1980, it was the only school that had a neuroscience department, even though it's a tiny school. Interestingly, Penn was one of the first schools to, along with Amherst, to make a neuroscience major. But at Penn, it wasn't a department. It was an interdisciplinary thing, combining biology and physics and math. Whereas Amherst, somebody gave a bunch of money to make a department in neuroscience, so that got my eye. So I focused right from the college on the interface between psychology and the biology of thought, and with an interest in schizophrenia, and so I never really stopped from then. At the end of med school, I wasn't sure what I wanted to do in terms of a PhD in neuroscience versus the more medical route. And so I teched for a year in the Connecticut Mental Health Center in a biochemistry lab, really more epilepsy research. And I got a couple papers out of that on GABAergic inhibitory synaptic transmission and epilepsy. And I also volunteered at what was then called the Yale Psychiatric Institute with adolescents with psychosis who were on an inpatient unit. And my volunteering was to try to be semi-normal person for them to interact with. So I played chess with this kid that was actually he was a senior at Trinity when he developed severe schizophrenia and paranoia. He could hear the voices of, he was a philosophy major and a kind of religious studies guy, I think also. And he could hear the voices of Gabriel, the archangel, and the devil screaming at each other and fighting in his head. He also was a chess champion. And I was okay at chess, not at his level, except I could beat him when the voices were bad enough. But when they weren't, I had no hope whatsoever. But so that was played chess with this guy and kind of learned a fair amount about what it's sort of like, to have this going on in your head, but yet be kind of trying to be normal. And I worked with a number of other young people there. I did SAT prep with a number of people there. And that got me even more interested because I got to work with people that are in that transition. And just by luck, I was living in a house like you do right after college, right? So, lived in sort of a boarding house, because I was teching and you don't make much money doing that. So where you have a room and it's, and woman owns the house and bathrooms down the hall and there's a few kind of boarders living in there. And it just happened that her nephew, had developed schizophrenia. So it's this weird coincidence since I was already interested, but he couldn't go home. Stuff had happened and that wasn't an option. So after 2 months in the hospital, when he first got out from his first break, he was the kind of, proverbial, pretty challenged young man in the attic, and most days were pretty good. Sometimes you could hear him screaming. Other days he was pretty good, and we'd go running together. And I remember watching MTV with him once, and there was a lot of weird stuff happening. The early days of MTV, whatever this is, like 1986 or something, 84, 85, and you could see sort of trails happening from people's hands and whatever they were doing. And he looked at me and he said, "Is that me or is that actually on the screen?" Then it kind of occurred to me that he kind of goes through life thinking that, he's not stupid, and he knows that some of his experiences, both auditory and otherwise, are not experienced by others. and he's just sort of reality checking, because he knew me and sort of trusted me, but that also was just something that kind of reminded me about what it's like to live with schizophrenia, where we tend to like to wall them off as if they're like not human, because it's kind of easier for us than trying to appreciate how much pain they're in. But when you get to know them, like, they're human, and they often have very weird sensory experiences, and from that develop delusions and paranoia. And but at the same time, they're lonely, they care, they have sexual interests, they're people, and I got a real bird's eye view of that by living with somebody going through it. But also, again, it was volunteering, so I got immersed in that world. I decided to go the medical route, partly because at the time, in the mid 80s, a couple things were going on. One was we had a government that came to power the concept that if we cut taxes for wealthy people, there'll be job creators, and the economy will grow. What ended up happening with that, and what's happened every time that's been tried since, and I think it's been tried 4 times since, and each time the same thing has happened, is it enhanced the recession, and the government actually does do useful work some of the time, including support the NIMH. So the year I was teching, the NIMH was at an absolute free fall. The core grants from NIMH were under 5%. So I knew PhDs that were going to teach high school, which is great if that's what you want to do. It's a totally awesome path. But if you thought you were going to kind of have a lab and stuff, there was no jobs because there were no grants, because the budget deficit went crazy because the taxes were cut. And the economy took a while to recover. Eventually it did. But at any rate, partly because of that, I went the medical route, but it was also partly because I was interested in learning more about treating people with schizophrenia. But I never stopped wanting to do research. And on my application, I only applied to one school, my University of Connecticut, and I grew up in Connecticut because it was a state school. I could live at home. I had a job during med school, and so I could get out without a bunch of loans. And you really couldn't do the path I followed unless you were really wealthy, because the typical med student these days graduates with a couple $100,000 of loans, and that wouldn't be doable with the kind of research track I took. I wouldn't be able to. It would take me 30 years to pay off those loans at a research salary, and seeing patients, but not enough patients. And also schizophrenia patients don't reimburse that well relative to other things. Most of them are Medicare patients, not private insurance. So anyway, that's the route I took. I did research every summer I had in med school, and different types of research. Some like evoked potentials on kids. So I was interested in brain development and how you'd measure that in kids, and evoked potentials are one way to do that. And in a couple stints at the NIMH, and then I also, I don't know, I took some time off because I decided I wanted to find out what being kind of a real doc is like, knowing I was going to go ivory tower. And I spent 6 months in Guatemala, 3 months in Belize, and 3 months in actually a migrant farmer public health clinic in Central Florida. And I can tell you that, that wasn't as much difference between the mountain village in Guatemala and the clinic in Central Florida for a lot of the resources we had. We had kids in Central Florida in the mid 80s dying of diabetes because there was no access for insulin. And things are different now, I think, to a large extent, but at least at that time, it was pretty rough sometimes. And it was obviously rougher in Guatemala for kids with severe challenges, but and there was also a US-backed counterinsurgency thing that was going on, war going on at the time. But anyway, so I came back, finished med school. I went to Pitt for my psychiatry residency. At the time, it was one of the 2 most biologically oriented psychiatry programs. Some of the Ivies, including Penn, were a little slow, partly because they kind of had the old stodgy professors more shrinky oriented, more Freudian oriented, still in power, whereas some of the younger schools or less prestigious schools had made more of a biological switch a little earlier. I would say that's not at all true today, at all. I mean, actually, Penn is one of the world leaders in mental health research and psychiatry, and is very biological. But there also is a lot of important evidence-based talk therapy that does help people. And Penn's been a leader in that. So at any rate, I went to Pitt. Did a lot of schizophrenia research, worked in monkeys and some humans. After that, I had to decide what to do. I had a couple publications at Pitt, but and a lot of people were getting into brain imaging, and I had a chance to do that. I did a little bit of it, but MRI was just starting to be invented then, and MRI spectroscopy and also functional MRI. So ways to look at what part of the brain is more active were available. I had actually done a summer fellowship at NIMH in PET scanning, which is a radioactivity-based method for measuring blood flow changes, but the scale of the temporal resolution of PET is really low. You have a radioactive tracer in the blood, it kind of comes in, it binds, and then it leaves. It takes minutes to tens of minutes to see a change, whereas functional MRI, you can see changes in seconds. So it was a big game changer, but I still felt like we didn't know enough about the sort of core aspects of how the brain develops to use imaging to really understand what's happening. Ultimately, when we come up with therapies, they're going to be based on molecules, and not on pixels. And you can, and especially with spectroscopy, you can measure molecules with pixels. And I might have time to mention a little bit about that later. Yeah, in an arrogant kind of way, to me, it felt like smart people were asking stupid questions using some of the new technology, because we didn't know enough about how the brain actually develops and works at a more molecular level. So for me anyway, I went away from what I had done during residency and did a postdoc at UCSF in developmental neurogenetics. That was the early days that people have developed techniques to make knockouts of genes. And so you could, pick a gene, knock it out, study what it does with one copy or no copies in a mouse. And of course, mice don't have schizophrenia, but mice do have behaviors and alterations in cognitive abilities that you see in people with schizophrenia. And sometimes you can find those occurring when you alter a gene that is known to be a risk gene for schizophrenia. So you can do some useful work there. And that's what I did. And I didn't focus on schizophrenia at all. I focused on development of inhibitory interneurons, the cerebral cortex. There was reasons from what I had done previously and what the literature was showing that these inhibitory interneurons are important in schizophrenia, but we knew nothing about how they develop. And so I wanted to go to a lab where I could study how do these cells develop. And I kind of got lucky in a science career. This is actually, people often don't talk about it. And, I'm not dumb. I'm reasonably smart, within the full professor, kind of made scientist folks. But, I know people that are smarter. I certainly know people who are successful I don't think are maybe as smart. But you end up to kind of make a break. You kind of have to be persistent, and there's a certain element of just luck. So I wanted to understand how these inhibitory interneurons develop, and I was working in a lab, and I chose a lab because I thought maybe that would be the place to go. But I also got pretty lucky. They happened to be, well, I knew they were working on gene knockouts of transcription factors. So these are, genes whose proteins control the expression of other genes. They tell genes when to turn on, when to turn off, how strongly to be expressed. And so I wanted to study those genes because I thought I could learn, how the forebrain develops and then maybe eventually how interneurons develop. And just really kind of by luck, they had been working on making a knockout of two genes that are very closely linked. There was a a duplication event about 150 million years ago, and one gene became two. And once that happens, they often start mutating because the mutations are tolerated because there's another copy there. And so they drifted away from each other some, but they're close together. And they both are related to the striatum development in the sort of ventral part of the brain. And so we started studying that knockout, and I was kind of lucky because I was more of a neuroanatomy person, so I kind of knew something about how to study how the brain develops. Or more of the people in the lab were gene jocks. They could clone in their sleep, and they could, work on this. I helped with the cloning and with validating the molecular side, but I was like, a babe in the woods relative to these people did their whole 7 years of PhD on like hardcore molecular biology. But I did know about how the brain developed somewhat. So anyway, I got lucky, and we happened to make a knockout that the genes that were deleted were in the ventral part of the neural tube. And if I could sort of just do this, yeah, I could see. So you have a neural tube during development. The cerebral cortex in the forebrain is up by my fingers, and then the ventral part of the neural tube by my thumbs. That's going to make the striatum, the basal ganglia. And so when I was doing this stuff, the prevailing model was that the cells are born on the inside of the neural tube, and then they migrate radially on radial fibers to go out to their position in the developing, whether it's cortex or basal ganglia, anything. The nice thing about that system is like kind of spokes on a wheel. And the cool thing is that way, if you want to study the genes controlling, at least this directly controlling the cells developing in the neocortex, you only need to study the genes up here in the top of the tube. Clearly they're connected to cells that are other places, so they're going to be influenced by other things. But at least the genes that are directly related to their development are going to be in the same part of the tube. So what I was able to show, because we made this knockout, that eliminated sort of the part where my thumb is, or changed the way. And then what we saw when I looked at the cells in the developing neocortex, the inhibitory interneurons were like gone. And this was sort of completely contrary to the way people thought the brain developed. It wasn't appreciated that cells could migrate across the radioglial scaffold in this way, and particularly that they wouldn't be such a prominent group of cells. So the neocortex has excitatory interneurons that are about 80%, inhibitory interneurons about 20%. The two have to work together in order for the balance of excitation and inhibition to be right, and also for the interneurons actually synchronize. They're a little bit like the conductors of an orchestra. They synchronize the activity, not just inhibit it. And the synchronization is actually how our thoughts are the neural mediator of consciousness isn't in the individual firing of neurons, it's in the synchronous firing of neurons. So the interneurons aren't working right, fundamental concepts and fundamental aspects of consciousness aren't going to work right. So anyway, it turns out the interneurons, they come completely from the ventral part of the tube and then migrate up. And so I kind of got lucky. In the same year, in 1997, I had a first author paper in Science and a first author paper in Neuron on this stuff. And that kind of put me up as like, wow, this rising superstar. But honestly, a good chunk of it was luck. And I worked hard, and I was already interested in like, how do these cells develop? So my myopia probably helped, but there was certainly a component that, I placed a bet and it paid off, and it didn't have to, could have studied some other gene that didn't have any phenotype. My boss was trying to get me to study the gut development, because the nervous system in the gut was also altered in these animals, and I had to kind of fight him off. And I was lucky that I had a mentor who would be like willing for me to be able to say, like, I don't want to study the gut, I want to study the forebrain, so I had to be persistent in what I'm interested in, but I got pretty lucky. So after that, I decided that a particular aspect of it, I kind of wrote a couple grants, got grants on how the cells migrate, but I wasn't as interested in how they migrate as in how their fate is determined, because the cell fate aspect of it to me was really interesting, because normally like in the retina and the neocortex and lots of parts of the developing nervous system, you got to figure out how many types of cell type A and cell type B you want to make, right? You got a bunch of different neurons. How many of each type do you want? And so usually the developing field feeds back on the proliferating cells to say, hey, I got enough cell type A, start sending me cell type B. And that's, you can demonstrate that in the retina and in the neocortex is a piece of how neuronal diversity occurs. So that's impossible if the inner neurons are coming from a completely different neurodevelopmental domain and doing this long tangential migration. You'd have to have a completely different system for coming up with how you're going to make the right number of the right type of inner neurons. And the types of inner neurons are quite diverse. Some of them mainly inhibit other inner neurons. So if you lose them, the circuit becomes hyperactive. You can get epilepsy by losing inhibitory neurons because they're the inner neurons that inhibit other inner neurons. Whereas others are very potent at stopping or sculpting the activity of the excitatory cells in different ways. Some of them project mainly to the dendrites of the pyramidal neurons. Others go after the cell body or even the axon digital segment where the action potentials are forming. And so to me, I got really interested in the diversity. So I did that for about 10 years at Cornell Med. That went well. About 2/3 of the way through, the human stem cell thing started happening. I'd already been working in mouse stem cells for a number of years, but this gave us the opportunity to start studying how these genes are regulating development of actually human cells. And human cells are not necessarily the same as mouse cells. There's a ton of, there's an amazing amount of similarity in forebrain development. And, and back in the 90s when I was a postdoc, we were joking about how we're all flies because a number of labs had shown that you could take a certain transcription factor, clone the gene out of the fly, knock it out in the mouse, put the fly gene in, and the mice are normal. So like 200 million years of evolution, the same protein does the same thing in developing, of course, in the brain of the mouse and the brain of the fly are not at all the same, but they'll still rescue the function of that gene. And the forebrain development of the mouse would be normal with the fly gene inserted. So, there's a tremendous amount of conservation, but in humans, there are also a lot of, there are a number of genes that aren't so well conserved, but also there's processes that are just different because our brain is so much bigger. The duration of development, both prenatally and postnatally, the human is so extended that you need to, the opportunity to study human cells got really interesting to me. And so I kind of dove into that kind of in the second, the third wave of the stem cell thing in the late 2010s with a wonderful collaborator, Lorenz Studer at Memorial Sloan Kettering across the street from where Cornell Med is in Manhattan. So then I decided what I really wanted to do then was change my career trajectory. Things were going well, and I was publishing papers in the top journals relatively frequently, but I wanted to switch to doing more real clinical translational research. Being a physician scientist, I actually ran a psychosis clinic at Cornell Med when I was there for 10 years. It was focused on treatment-resistant schizophrenia. And I wanted to see, can I actually use some of this knowledge to see if we can help them, at least add a little more focus on the translational side. And I don't know whether this necessarily makes any sense because we have so much to learn about the basic development that, I still wonder whether we're asking stupid questions because we just don't know about how these genes actually work. And we could study that in flies and worms and mice and learn a ton, so maybe it's a little premature to go to the human system. And plus the stem cells are, you can't model adolescent era development when schizophrenia develops during adolescence for a reason. There's a ton of synaptic pruning happening in adolescence. And it seems that there's something wrong with the pruning or that the pruning is unmasking a deficit that was already existent but didn't have a phenotype until after the pruning occurs, and now you have less synapses and they're not working well, and then you run into trouble. So, I'm not saying that I think it could have made more sense to stay with a more basic system, but but I decided I wanted to take a shot at doing more translational work. So I moved to Penn 10 years ago because of two things. Doug Wallace, a mitochondria researcher, who is, he's one of the guys, he actually was one of the people who discovered in the late 70s that mitochondrial DNA is maternally inherited. And so, he's kind of my hero. He was literally on the cover of National Geographic, right? I mean, that's pretty good. And it's one thing to have like the molecule of the year at Science or Cell or something, but so he created some of the early maps of the out of Africa using people would say the genetics of Eve, and using mitochondrial genetics to track migrations of humans out of Africa across the planet, and has continued to do amazing work in mitochondria. And so I wanted to position myself between him and between a woman at Penn, Raquel Gurr. So Doug's lab is at CHOP. He's also a Penn professor, but he's children's hospital based. And Raquel Gurr, and she studies schizophrenia, and is a world leader in schizophrenia research, more on the phenomenological side. Like, how does it develop? Is it different in men and women? How is the brain changing during the development of schizophrenia? She was really focused on that early stage of like, are people who are going to develop schizophrenia different before they develop schizophrenia? Are there ways we can use biomarkers to identify who's at highest risk? And I had reasons to suspect that mitochondria could be important, and wanted to kind of wedge myself between the two of them. And then, even though I hadn't really studied mitochondria before, and then see if I could, connect the dots, as it were, and still thinking about forebrain development and my inhibitory interneurons and stuff like that. And so that's essentially why I came here a decade ago and started focusing on that. And I can say it took some time, but it's going pretty well now. We have been able to identify that in some instances of schizophrenia related to this particular genetic alteration that CHOP has the world's largest clinic of, called the 22q11.2 deletion syndrome. So it's 46 genes and 13 non-coding RNAs or so deleted on chromosome 22. And they have about a 25% risk of developing schizophrenia. And there's also a pretty good mouse model. So my lab's been working with the mice and with iPSCs from people with 22q deletion syndrome who either do or do not have schizophrenia. We had reasons I won't go into here, but if people want to hear about it, they can ask. I can go on that stuff forever. But on mitochondrial deficits in those both the mice and in the iPSC derived neurons, stem cell derived neurons from the individuals with schizophrenia versus without, we found that the ones that have the deletion but don't have schizophrenia, their mitochondria have compensated for the effect of the deletion, whereas the ones who do develop schizophrenia, they fail to compensate. So the funny thing is the gene expression in some extent is actually more abnormal in the ones without schizophrenia than the ones with schizophrenia, because the gene expression changes in the ones without to do this compensation thing that the ones with schizophrenia somehow fail to do. So we're trying to figure out why they fail to do it, because that might be a treatment mechanism, or why they do compensate, because that might be a treatment mechanism. But in the meantime, we also have an FDA approved drug, or actually a whole class of them that are approved for hyperlipidemia, that activate generating more mitochondria. And they're well tolerated, and they're used in adolescents quite a bit, including at CHOP. And so we use that medication, we put it on the stem cells, and we normalize the abnormalities in mitochondria and some other things in these stem cell derived neurons from the plus schizophrenia group. And now we're trying to see if we can go through the process, which will take years, to get the FDA to approve an investigational drug trial, and so that we can see whether we can actually prevent the development of schizophrenia in this special case of 22q deletion syndrome. As I said, they have a 25% risk. We published a paper in JAMA Psychiatry last year where we showed a method where we could have a biomarker that we can measure in their blood cells for which kids, before they develop schizophrenia, have either compensated or not. And so if we can identify the ones who have not compensated and show that they seem to have like a mega risk for schizophrenia, like 80%, now you have a chance with a preventative trial to show like even if you have a small trial, like 20 kids, but they're selected for the ones that have an 80% risk. If you treat them, you might be able to show that now only 10 of them develop schizophrenia, whereas you would have predicted that 18 do, or something like that. So we're trying to work through all that part. And so things are going well from the perspective, this wouldn't be all people with schizophrenia, but probably more than just a 22q situation, because there's reason to think that the mitochondria are important beyond that. But that's a little bit of a really quick nutshell of sort of how I got where I'm going now. And then maybe we can stop here for questions.
Actually, well, there's one question in the chat about the drug you've just described. So the question is if this drug is going to help the mitochondria to compensate.
Yeah, so the medication that we're looking at, the specific one, again, it's a class, and there's a bunch of different companies that sell different versions of it that are slightly different from each other in a variety of properties. But we've been focusing on a medication called bisofibrate. It is an activator of a transcription factor called PPAR alpha. PPAR alpha is a transcription factor that will activate several hundred genes, both in the nucleus and the mitochondria themselves. They're kind of like little bacteria in our cells that specialize at generating ATP. And so they have 13 genes in the mitochondria that also get activated by this PPAR alpha. So the transcription factor drives both nuclear genes that are needed in the mitochondria. There's like 1,100 proteins in the nucleus that are needed for mitochondrial function. And there's 13 in the mitochondria. And so PPAR alpha drives both groups, and bisofibrate then will activate more mitochondrial biogenesis. So it's used for hyperlipidemia, when you have too much fat in your blood. And when you crank up more mitochondria, they basically can metabolize the fat and lower your fat levels. And so that's why it's a treatment that's used. Any other questions on that? What I was going to transition to was more on the shadowing side of things, showing you a little bit like sort of what's on my desktop, like what kind of things do I work on during the day. I was actually just going to go through my schedule and just show you guys, like, this is like what I do, by showing you my schedule for like next week and the week after, something like that.
That would be great. There's just one more question on the treatment side of things. So the question is, could epigenetics play a role in which people are able to compensate with altered gene expression and which don't?
Yeah, totally. Chris, the word epigenetics, I've always been a little confused about how to translate that word, because it seems to basically be used for control of gene expression in common usage. And then we used to just use more words and say, like, control of gene expression. The original definition of epigenetics was a heritable alteration of gene expression that was not in the DNA. And so the word heritable was a really important aspect of that. And some of the first people that studied epigenetics were actually a group of Russian biologists who had noticed that when you capture mink, and this was done in the 20s and 30s, and also fit within some aspects of communist philosophy, interestingly enough. So that was partly why the state supported it. But if you take mink and you capture them and you house them in in cages where you're breeding them and you're feeding them, their coat within a generation or two will change and start becoming whiter instead of browner and mottled. And also their behavior changes, and they learn to be more comfortable around humans. And that learning seems to even occur in a way that's not something that's learned. Like, you can take the offspring away right away and they'll still behave differently if they're a second generation than if they're like, if you take a pup from a wild mink and you grow it in a cage, they notice that that pup will be more wild than if you take a pup from a pair that had mated in captivity. And so this was some of the things that started. Their genetics are the same, but something's different. And that sort of started some aspects of the field of epigenetics. Ties in with communism because of the challenge. Like if you're going to remove greed as a basis for society, how is that first generation going to change to accept a system where greed is not the basis of societal reward? Because if they grew up greedy, they're going to be greedy, right? And that's a challenge that Marx dealt with. So they love this work in the Russian Lenin system, but also they did some good science. And so I originally learned epigenetics in that context, a heritable, non-DNA based alteration of gene expression. But it's now used to basically mean gene expression. So now to get back to your question, yes, epigenetic changes are absolutely critical. And so to give you a really quick snippet on an experiment we're doing related to this, and I'm going to stop sharing. We have a hypothesis that I can't go into in the time, but that the communication between the mitochondria and the nucleus are part of the reason for the failed compensation or the presence of compensation. To test that between the 22q kids with or without schizophrenia. So to test that idea, we're harvesting mitochondria from blood cells from kids with or without schizophrenia. And we're transferring those mitochondria into stem cell derived neurons from people with schizophrenia. Our hypothesis is that the mitochondria from the kids with schizophrenia are not going to improve the metabolics of the neurons from the kids with schizophrenia, as well as the mitochondria transferred from the kids without schizophrenia. But more importantly, and that's relatively easy to measure, but our real hypothesis is that if we can see that effect, which would be cool, we actually think, or at least I think, that the effect won't be because the transferred mitochondria are like juiced up and they're cranking out a lot more ATP themselves. I think it's going to be that they're changing the epigenetics of the nucleus, because the mitochondria communicate with the nucleus, and it's that communication which I'm hypothesizing is at fault in the reason why 25% of the 22q kids don't compensate, and those seem to be at higher risk. So we can come up with that mechanism. There are a bunch of non-coding RNAs in the mitochondrial genome, and some of those might be regulating this like message back to the nucleus. Hey, I need a little more love here. I need more proteins to grow more, and so that's what we're working on, related epigenetics. It's a really good question. We'll see whether it pans out. It's definitely high risk, high payoff kind of stuff in this case. Okay, so. Yeah, any other questions? That was a really good one.
Not so far.
All right, so then what I was going to show was, let me go back to the share screen, a couple of just sort of the practical things that I kind of do. I'll show this one for starters. Some days, I'm sitting in my desk and I'm studying stuff. And so this is a spreadsheet of de-identified information from individuals that have 22q deletion syndrome and are with or without development of psychosis-related symptoms. And so this is part of a NIH-funded study that I'm the PI of, the principal investigator of, and we're trying to see whether we can use the blood cells to predict who develops schizophrenia. So the clinic at CHOP has been banking blood cells from kids for like 20 years. And many of them, they have follow-up that goes for a decade or even more. And so we're identifying the LCL, these lymphoblastoid cell lines. So they're immortalized. It's sort of an issue potentially, but we've already showed that they can be useful. Many people have, before me and my lab, showed that they can be useful for studying mitochondria. So we're trying to see whether we can identify the kids that either did or did not develop schizophrenia, and then study their LCLs, their blood cells, and see whether we see a mitochondrial compensation or not before they got sick, right? This would be critical for this drug trial, because then that could get us to what I had mentioned about something like 80% risk. And that would then, if you're going to try to prevent something to happen, you need it to happen often enough so that with a reasonable number of subjects, you can actually show an effect, right? So that's something that we're trying to do there. So that's, like I pour over the spreadsheet, picking the ones that I think we should use. I'm working with a postdoc on it, but this kind of thing I'm kind of doing myself, because you got to make sure we get it right. It's very expensive, and let me see why I didn't get the rats. All right, let me just close this one. I don't understand. I'm clicking on what I see as being the one I want to show, but it's not showing. All right, if I close that, then, all right, so another spreadsheet I'm looking at. This was prepared by a technician in my lab. We're studying, and I'll allow this t-shirt from the CHOP epilepsy program, where my lab's helping to identify why certain kids develop epilepsy by studying stem cell derived neurons. Another lab, Ethan Goldberg, who's actually the leader of this project, he's an MD, PhD child neurologist. So in this system, my lab is more the technicians, we're just making the cells, and they do the fancy electrophysiology and imaging on the cells to see whether they can figure out the mechanism for these epilepsies in kids that are pretty rare, but common enough so a tertiary center like CHOP sees quite a few of them, to see if we can develop personalized medicine to help them. And so in this specific thing, this is the differentiation of kids with the SCN3A is a sodium channel mutation. It's the 7th differentiation. We're using a particular line that's a CRISPR mutant and a corrected control. So the line is from a control, and then we mutated it to have the deletion seen in the kids. We have other lines that came from the kids, and then we fix the deletion to make it a control, and that way we have two types of comparisons between mutated and control. And at most of those, my lab is in some of that, but most of these were done in the core facility at CHOP, not in my lab. So this is just an example. So he's got to go through the spreadsheet of the date where everything's supposed to happen, what kind of media changes, and you got to feed them, and don't add the ROC inhibitor and the different factors. And so he sends these to me to review, and just to make sure that we're actually doing what was the plan. And so that's something that I do pretty often, is just look at the plan, and then study, see whether it's right, or of course what I think is right may or may not actually be right. Something else I often do is review papers. So this is a paper that I'm reviewing from another lab. It's on interneurons and their impact on psychiatric disorders. And another one that I had brought up, I was reviewing a grant from just this week on vacation from this group. And let me get back to Zoom. Where's Zoom gone? And so I'm reading a grant, but to read the grant, I have to read some of the group's papers. And so transcriptional programs regulating neuronal differentiation are disrupted in DLG2 knockout human embryonic stem cells and enriched for schizophrenia and related disorder experience. So, in order to understand the grant, I got to do a little background reading and get my head around the research program. And of course, in the process, it's not just reviewing the grant. I also learn a lot, that's a key part of science, is this peer review, where we, who else could review papers or grants but people that are in the field, because it gets so specialized, and you hope that people are ethical and don't steal things from each other. And I think the vast majority of the time that's the way it is, because we all have sort of special little niches for the most part, and so it's usually not evil and competitive. It's usually people try to help. If they see a flaw in a paper, they say, yeah, I think I see where you're going with this, but I don't think your conclusions are reasonable based on the actual data you have to support them. I have this other way of looking at it. Can you help me understand why I'm wrong in thinking that you're not seeing it in a way that's sort of fair, so that's a key component. All
Actually, I
right,
got a
and
question related to that. So the question
cool.
is it common to collaborate with other labs facilities for such projects? What other important collaborations are you currently doing?
Yeah, great question. And actually, I'll even jump to my other share screen here and sort of show you what my week looks like related to that. Um, so tomorrow, first I have an hour and a half meeting um, where because I'm a leader and I have to be trained in leadership. This is kind of painful, I must admit, but I have to do it. Um, it's more a McKinsey and corporate leadership model, and obviously, CHOP is a corporation, and so I have to go along with the servant leadership thing. Their model doesn't really fit within the teaching mentorship framework. It was designed in like IBM or AT&T or whatever, big banks. But I do have to go, and I'm learning to be a servant leader. And then I have a meeting, this one with Bill, Stu, and Thomas. So Bill's a postdoc in my lab. We're collaborating with Thomas DeRate's lab at CHOP. Thomas studies glioblastoma. So these are terrible tumors that develop. They're one of the leading causes of death for kids that are non-injury related and not car accidents or guns. And so I saw a talk by this guy, and he's talking about these secreted factors that the glioblastomas release, and those factors cause the immune system to become more quiescent, so that the tumor kind of learns in a way to grow without being overly attacked by the immune system. And those are the tumors that are successful at spreading and killing. And so I'm watching this talk and I realized, well, wow, those factors are the same factors that guide interneuron migration during development. So I bet if we transplant interneurons close to one of these tumors, they're going to go after it. If we transplant interneuron progenitors, like just after they're born, we can harvest those from mouse embryos, and we can also create them from human stem cells. And so we got a small pilot grant from CHOP to start those experiments, and they went really well. And then we got a DoD, Department of Defense grant, and I think Thomas just told me while I was on vacation, he got 2 more grants related to this idea. It's a pretty cool idea. We have some pretty strong evidence that it can work for some tumors, and we're now modifying the migrating interneurons with something related to the CAR T technology. That's a big deal. Carl June at Penn will win the Nobel Prize almost certainly for this technology. And just for fun, Penn and Vanderbilt are fighting over who actually owns this cow, because Carl was at Vanderbilt, where some of the initial technology for that CAR T thing was developed 20, 25 years ago in his lab. Anyway, we're trying to see whether we can use the interneurons, since they'll migrate to the edge of the tumor, as little missiles to bring in an immune activator to then tell the native immune system, get these guys. And so that's what this collaboration, and we meet every week about that, is about. So that's one example of a collaboration my lab's doing. I mentioned the SCM1A meeting collaboration with Ethan Goldberg, and I have a weekly meeting with Nate Satuyo. That's this one, where we're going over in this case, we're using a cell-based therapy. We're transplanting interneurons so far from mouse embryos, but we have a grant from the Eagles Autism Foundation that kindly supported us to try to see whether in this model where there's both epilepsy and autism, can we use interneuron transplants to fix one or the other or both, depending upon which cells we transplant and where we transplant them in the brain. And so we have a weekly meeting about that, and that it's a very collaborative project. And then another collaborative project on my weekly schedule is here with this Levy Genomics. So this is the Lifespan Brain Institute. I mentioned Raquel Gur was one of the key reasons I came here. She's the director of this institute that CHOP and Penn co-funded. I'm the associate director. And it's really designed to try to integrate some of the developmental neurobiology related to mainly to schizophrenia, but also other neuropsychiatric illnesses across CHOP and Penn, because we have different. CHOP is an independent institute, so although my paycheck says Penn on it, I'm a Penn professor, all my, CHOP owns my brain, they own my intellectual property, and also the compliance stuff. So my grants run through CHOP. So it's in my IRB, institutional review board that approves human subjects research, separate at CHOP and Penn. Animal committee, separate at CHOP and Penn. So our Alibi is designed to try to break down those barriers that sometimes form, both from the suits who are, doing their job and protecting intellectual property, and also the compliance stuff with IRB and animals, so that we can facilitate work between the two institutions and really focus on how can we help the kids. And so we have that meeting. We have a bunch of different meetings related to that. Some are about treatment, and I've discussed the bisofibrate idea that with this group. We also have some genetics related studies going on together, and a variety of collaborative grants. So that's another big collaboration that I have. I have one more that was on here. I think this one that was canceled was another collaboration. So Kelly Jordan Shudo in the dental school. My lab, I had a student who really wanted to go do his PhD and then go into industry. And so for his theme, it was sort of frustrating. He didn't care so much about testing hypotheses. He just wanted to generate as many different cell types from human stem cells as he could and get credit for that. So he would market himself to be a stem cell guru for companies. And so, I was like, down with that. So we made a really nice protocol for generating microglia. They're kind of the macrophages of the brain. So we started making microglia. And then I'm like, okay, well, who wants microglia? I kind of want to study them because of schizophrenia and synaptic pruning is related to what microglia do during adolescence. So I'm interested in that. We are working on that in my lab, but low-hanging fruit wise, and because as a scientist, you got to pick your battles. You can't compete. So I'm new in this microglia field. I didn't want to just run a microglia grant because the microglia people will be very suspicious of me. I came out of nowhere, and I'll get slammed. So instead I reached out to Kelly because she's an HIV investigator. Microglia are the cell in the brain that gets infected by HIV. Now you say, Kelly, I think my lab's making some pretty good microglia. We can grow them in a dish. Do you want to infect them with HIV? We can study whether how the HIV is affecting the human microglia gene expression. We can do treatment related studies. We can do studies of when they go latent, which is a key component of long-term AIDS. And so we have two RO1s. They're Kelly's RO1s. Again, I'm sort of a technician on this one. And I'm using it to get cred that I can use to get my own grant, really more related to schizophrenia in a way. And of course, hopefully we're doing some good science. So that's another collaboration that I have with Kelly in the dental school at Penn. All right, any other kind of questions on that? So some of the other stuff I have on here are the meetings with individuals in my lab. So I meet with Bill after we meet, he's a postdoc, after we meet with Thomas. On Fridays, I meet with a technician in the lab, Kiona. She's studying cannabis. They also affect microglia. We have this idea on related to schizophrenia. There's evidence that schizophrenia risk is increased by cannabis use, but only before you're 16. That evidence comes from 3 different studies on 3 continents. So it's pretty good evidence that that's a real thing. So we're trying to figure out why would cannabis cause schizophrenia. Actually, cannabis causes microglia to become more quiescent. There are CB1 and CB2 receptors on the microglia, and both of them tend to make the microglia more mellow, less likely to kind of be snatching synapses. However, I started thinking, well, how do kids use marijuana? They tend to use it in the afternoons or early evenings. Maybe they go home, then they, have dinner with their families or whatever, and then go to bed. All right, so that means that if the cannabis is suppressing microglia activity late in the day, and the release of that suppression is occurring when? While they're sleeping. Well, while they're sleeping, the circadian rhythm is such that the microglia become more active in synaptic pruning. It's more at night that this pruning thing is happening. So if at that time when their circadian rhythm is that they're supposed to be actively pruning, and now they're sort of hyperactive because they're rebounding from the suppression caused by the cannabis. So basically with immune cells, they're very reactive. If you push them down and let go, they bounce back, they overshoot. And there's tons of different papers showing that happens. So my idea is that they're going to overshoot during sleep. What's that going to do? It's specifically during sleep. It might make them hyperphagic for the synapses, maybe enhancing this overpruning thing that is pretty good evidence can be related to schizophrenia. So we're trying to model that in the dish. And then with Mariella de Blasio at Penn, she has a lab where she's got mice that are vaping THC or CBD or other cannabinoids in a closed system. And so we can vape the the adolescent era mice and then study whether if we vape them with cannabis late in their day, which is flipped circadian wise to us because they're nocturnal. Then we study their brains after a week of vaping every afternoon for them and see whether they've overpruned. This would give us some mechanistic idea about connections between cannabis and schizophrenia. So that's what that part is about. So yeah, so that's a little nutshell. Hope you guys get some sense of the shadowing. I didn't mention, I stopped seeing patients when I moved 10 years ago to Penn. That they wanted me to take a more administrative, leadership kind of role. And, in my line of work, there are a lot of psychiatrists that do basic science research and publish scholarly papers and stuff. So by the time I was a full professor at Cornell, I was getting nibbles from people wanting me to come and be a chair of a department. And I never applied for any of those jobs. I'm not saying I would have gotten them anyway, but I didn't apply. I wanted to be a scientist. But in taking the position here, some of the stuff like I'm doing with the Lifespan Brain Institute with Raquel, it's a fair amount of administrative work. We have junior professors, we're hiring and we're strategizing on strategic plans. We're meeting with deans at CHOP and deans or the equivalent of deans at CHOP and at Penn. We're getting substantial support from both sides. We're going to donors. And so all that kind of stuff is like not in the lab, right? But it's stuff that's important to me. We're trying to build something and I'm excited to do it. But I had to give up seeing patients if I'm going to run my own lab and then do more of this administrative stuff, more mentoring of junior professors, more recruitment. Recruitments take a lot of work, because you got to wine and dine people and then you got to negotiate. Then I don't really have any power. I'm an influencer, but I don't sign the checks. I got to go to the CSO at CHOP and Penn and get them to agree to what I'm hoping that they'll agree to, and so that takes a lot of work, so that's a big part of what I do. And so I couldn't keep seeing patients and do that. So I stopped. I miss it, but I like what I do. All right. Any other questions? People can ask questions directly.
One thing I was wondering about is because you mentioned a lot of drugs you're studying and the effects of it. And I was wondering if you were ever studying the effects of antipsychotics and how they would they work in the instances you're researching, or is it like a completely phylogen, a different presentation of schizophrenia that
Yeah, and it's a really interesting field, and I haven't done any research in that in antipsychotics. To me, the idea that we're going to find, I think the antipsychotics have helped a lot of people with schizophrenia, right? For sure. But there are a lot of people that have schizophrenia that the antipsychotics don't help very much, or they take the edge off, which could be big, allow them to live in the community, but, they're nowhere near their previous level of functioning. And a lot of people still hear voices, even, maybe a little more than half of people with schizophrenia will still hear voices, even if they take their meds. So I started focusing more on a different type of approach, which is if we can identify the circuitry that's altered, let's see if we can fix it directly. And I published a paper with a guy named Doug Coulter. He's an epilepsy electrophysiologist. He's never published in schizophrenia before. I convinced him to work with me on a study of mice with this 22q deletion syndrome. We had reasons to suspect that this ventral part of the hippocampus is hyperactive in these mice. I won't go through why. And I convinced him to do a study where we're going to correct the hyperactivity. The circuit's hyperactive because the inner neurons aren't working right, what I think, but the circuit's hyperactive for sure. And I think it's because the inhibition isn't right. And I say, well, let's put a virus in that carries one of these things called dreads. So the virus carries a dread that only expresses in excitatory neurons. And when it's expressed in excitatory neurons, it's a receptor that when you give the animal a designer drug that doesn't have any side effects, it won't go all over the body like antipsychotics and cause all sorts of side effects everywhere. It'll only affect this designer receptor, and then it'll downregulate the activity of those cells. So we showed that the ventral hippocampal circuit is hyperactive, and we corrected the hyperactivity. Even though we think the reason it's hyperactive is the inner neurons, we corrected the hyperactivity with a virus we injected into the brain of adults. And downregulated the activity in a titratable way. You feed them a different amount, and then or you can inject them with a ligand, and you downregulate the activity of Perelman neurons. And now we corrected several psychosis related behaviors in the mice, in adults, with a focal circuit based designer drug approach. Very different from the idea that you take the pill, it goes everywhere, and you hope that it has enough selectivity. But the fact is, the receptors in our brain are used again and again in different regions and different cell types, and not just in the brain, and so it's very hard to have enough specificity at doses that don't cause side effects. So yeah, so for me, I've shied away from for more of the antipsychotic related research, but some of the research I've done has been more the circuitry based stuff. I don't know if we're ever going to get that into patients. My guess is we will at some point, but it's going to be tough because you're going to have somebody with chronic treatment resistant schizophrenia. You got to convince them that we're going to inject a virus into their brain that's going to change a specific subset of cells in a way where then we feed them this medication that's selective. It's a lot of informed consent, right? And for somebody who's chronically psychotic. So it's going to be a hard sell, but the technology is happening. We're doing it in mice. Other labs are doing it in mice. We got scooped actually when we published a good paper in biological psychiatry, but we got scooped because while we were doing the study, a paper came out in Cell that did the same thing, and as we did. So it's the idea works, and other labs are doing this. So that's something that we've been doing, is more the focal gene therapy kind of approach in a way, rather than the what drug can sort of fix things but be selective enough to not cause off-target effects that make the drugs really shitty to take, which is where antipsychotics are at. But there is cool research done on new classes of antipsychotics. It's definitely, I'm not against it or anything. It's just, but it's not my thing. Maybe I'm just too pessimistic, but that doesn't mean I might be wrong. Yeah.
Yeah, thank you for that elaborate answer. I know we're very much over time, but we want to thank you very much for coming in today. I think it was very, we covered a lot of ground during like very short time, and I'm sure everybody enjoyed it very much. And yeah, we wish you all the best, and thank you very much for coming. Have a good day.
I hope I wasn't too all over the place on 3 hours of sleep and late in the day. So
no, it was great. It was great.
Well, it's another thing about being a scientist is you never stop being like a college student. So when I write grants, I have to stay up to 3 a.m, sometimes 3 days in a week, 3 days in a row, right? So it never really ends that way. So feel free to reach out to me if you guys have questions. Yeah. So
Thank you very much.
take care.
Bye.
Thank you so much.